Hello, St. Charles. Hi, Pirates. This is Mr. Russell here with the second installment of our Obj7 notes. This one is going to be over protein synthesis. Last time we talked about proteins and their structure and their functions, and then enzymes, which are a type of proteins. So now we're going to start talking about a little bit about DNA. Some will be some review, and then we're going to go into the process of actual protein synthesis. So, protein synthesis is, and we've already discussed this some, but it's the production of amino acid chains, which we call proteins. Okay, the term synthesis means to put something together to make it out of smaller parts. So, protein synthesis, we are making proteins out of amino acids. Okay, there's two parts to the process: transcription and translation, and those both sound pretty similar. But we're going to talk about each one individually. Today we'll talk about transcription. Next time will be translation. And those are two things you have to keep apart. The way I usually remember is that they both start with trans, obviously, but transcription has a C, which is next, and then translation has an L. C comes before L in the alphabet. That's what works for me. Anyway, basically transcription converts DNA, the information in DNA, into mRNA, which is basically a second set of instructions, and then later on, outside the nucleus, that mRNA gets converted into, or the information, I should say, gets converted into a protein. Okay, so the protein production pathway, meaning the pathway that cells go through to make proteins, and it's what we really just discussed. You have DNA, which is all this information is stored. It's like the cookbook. I use this example regularly. I'm sure you'll hear more of it than you would like to over the next few weeks. DNA is like a cookbook. It's got a whole bunch of recipes for different proteins in it. Okay, anytime you make new cells, you have to go through replication so that you can get exact copies of DNA into all of your cells. Okay, once that information is in the cells and you need to use it to make proteins, you have to copy it into mRNA first. We'll talk more about that later. And then that mRNA information is actually going to be used for translation, which is where we actually use it to make a protein out of amino acids. Here's a diagram that shows that. Excuse me. This is the nucleus. You can see here this darker spot in the upper left. We have a strand of DNA, which has been unzipped. And it's here. You can see there's something creeping down it, going in that direction. That is the RNA, the mRNA that's being copied from it. Then that mRNA is going to leave the cell, and you'll see it much bigger out here. And basically the ribosome is going to cruise down that like a train going down tracks and start adding on amino acids in the correct order. And then we end up with a protein, which is what's being built here. Growing amino acid chain means it's a growing protein. Okay, so a little review. Nucleic acids, we talked about two kinds, DNA and RNA. All right, both of them are long chains of these things called nucleotides, which is kind of a big, scary word, but it comes from nucleic, meaning they're in the nucleus. And as far as the tides part goes, I'm not exactly sure where that comes from. Anyway, they're both long chains of nucleotides, and each nucleotide has three parts: a sugar, a phosphate, and a base. The sugar and phosphate make up the backbone. They give it its structure, and then the base is where the actual information is stored. Okay, there are 5 possible bases. Those are adenine, thymine, uracil, guanine, and cytosine. And here are their atomic structures, or molecular structures, I guess I should say. They're all very similar, but they do have subtle differences. Okay, base pairing happens slightly differently in DNA and in RNA. In DNA, G always goes with C. They are great companions. And A always goes with T. They're always together. And another fun way to remember it all together is, if you start with your A and T first, it's all thugs get crunk. I can't take credit for that. I think one of Mr. Wright's students like 3 years ago told him about it. But anyway, it's a good way to remember it. So G with C and C with G, A with T, and T with A in DNA. Okay, and here we can see that same thing. We see our sugars and phosphates on the side. The D here, by the way, is for deoxyribose, which is the sugar that's found in DNA. So you'll see that on both sides, and then in the middle we have our A's and T's and C's and G's all paired up correctly. Okay, RNA is slightly differently. G still goes with C, and A goes with U here instead of T. and that's just a slight little difference. T and U are fairly similar bases, but one is used in DNA, one is used in RNA. And that looks like this. You'll see we have our sugar phosphate backbone. Here they use S for sugar, but it's talking about the sugar ribose. And then you'll notice they have little bases attached to them. You'll also notice that this is just a single strand where the DNA is a double strand. Okay, so differences between the two. This should be review as well from last time, hopefully. Sugars, you found in them deoxyribose, which is the D in DNA. It has one less oxygen, and I'll have a little model down at the bottom here to show you that. RNA has ribose, excuse me, and that's what the R in RNA stands for. So the sugars, those sugars are similar. It's really just one atom difference, one atom of oxygen. All right, what bonds with adenine in each? In DNA, it's thymine, and in RNA, it's uracil. DNA is double-stranded. We talk about it being like a ladder. Ladders have two sides. If not, then it's probably not a very good ladder. And RNA only has one side. It's a single strand. Okay, their primary purposes are also different. DNA just stores information like a cookbook. Okay, and RNA has a few different purposes, all of them involved in the actual production of proteins, taking the information from DNA and getting it to the ribosome so we can turn it into a protein. All right, here are your ribose and deoxyribose sugars. The red dots are oxygen atoms, and you'll see 1, 2, 3, 4, 5 here. And over here we have 1, 2 hiding back there, 3 and 4. So one fewer oxygens is where the deoxy comes from. All right, we have 3 different kinds of RNA that we need to talk about here. DNA, there's really just the one kind. If you really get into some really technical science, there's different kinds of DNA that you can look at. But for our purposes, one kind of DNA, but there are 3 different kinds of RNA you need to know about. Messenger RNA carries genetic information from the DNA to the ribosomes, and we'll talk more about that in a bit. Ribosomal RNA is what ribosomes are made out of. Okay, so it's actually what they are made of. All right, and there's a picture of that. These are both the 2 subunits of RNA. Okay, there's a large subunit on top and a smaller subunit on bottom. Okay, transfer RNA actually moves amino acids around, and that's so it transfers them. But we'll talk more about that in just a couple slides. But that's what a tRNA looks like. This is a 2-dimensional model, but you can see it's all A's and C's and G's and U's. Okay, messenger RNA in a little more detail. As I said before, this carries information for how to make a specific protein. So it's just the information for making one protein. Takes it from the DNA out to the ribosome where that protein can be made. All right, they are about 500 or 1,000 nucleotides long. So links in a chain, remember, nucleotides. So each mRNA is about 500 to 1,000 nucleotides long. Okay, you read these 3, I shouldn't say you, your cells read these bases 3 at a time, and every 3 bases is called a codon. Okay, and I'm not exactly sure where that name comes from, maybe because it's got code in it, and it's kind of a code that we're talking about that cells use with DNA and RNA. But we read 3 bases at a time, each one is called a codon, and you can kind of think of that as being the special code that's in DNA is using 3 bases at a time. Okay, AUG is always the first codon or the start codon in any mRNA. They always start with that, and it will always give you this amino acid at the beginning. It's called methionine, which is kind of fun to say, methionine. And a good way to remember this, the start codon is AUG. Well, school always starts in the same month of the year, at least here in Missouri, and that month is August. So AUG is where things start. Okay, there are several stop codons that would be the last thing on an mRNA, and that tells the ribosome where to stop making a protein at. There's 3 different ones, and I don't really have a good way to remember these, except that they all have a U and an A in them. But any of these 3 will stop it. They all serve the same function. I guess you can think of them as the 3 different punctuation marks that you can put at the end of a sentence, a period, a question mark, and an exclamation point. So any of those will show up, or one of the three of those will show up at the end of the mRNA. Okay, so here's a picture of that. We have our mRNA strand. You'll see it's single-stranded, all A's, C's, G's, and U's. And you'll notice that we have AUG at the beginning because we always start with that. Okay, and every 3 bases is 1 codon, so it's kind of like a sentence consisting of all 3-letter words. The fat cat ran and ate the wee rat. Yes, champion. Okay, these are the amino acids that it would give you. Okay, we'll talk about that later. This part's actually the translation, but this is mRNA. Okay, peptide bonds holding the amino acids together. All right, let's talk about ribosomal RNA quickly. It's the main component of a ribosome structure. It's about 60% of what makes up ribosomes. The other 40% is mostly proteins. They have 2 parts, as I said before, a large subunit and a small subunit, and you can see this picture here. I like the hamburger bun model because if you ever pay close attention to a hamburger bun, the pre-split ones, if you cut your own, who knows what they turn out like. Mine are always terrible. But on a pre-split hamburger bun, the top part is always a little bit bigger than the bottom part, and ribosomes kind of work out the same way. So these 2 parts are both parts of a ribosome. Okay, last kind of RNA we need to talk about is transfer RNA. And these are called transfer RNA because they transfer amino acids. They pick them up because they're floating around out in the cytoplasm, and they bring them back to the ribosome. I shouldn't say back, they may not have been there in the first place. They bring them to the ribosome where they can be added to this new protein that we're making. So they run off around the cell and grab the correct amino acid and bring it back so we can add it to this protein we're building at the ribosome. Okay, they transport amino acids to the mRNA. The mRNA is going to be at the ribosome. They have anticodons, which are called anticodons because their bases are opposite of codons, so they match up with them. They're complementary. We'll talk about that a lot more when we get to translation. They also recognize the appropriate codons on mRNA and bond to them with hydrogen bonds. And that's something we're going to get into more detail in the next video. Okay, here is more of an accurate representation of the shape of a tRNA. It's not fun colors like this, but it is this approximate shape. This one just kind of shows you it laid out 2-dimensionally, but you'll see that it's just a long string of nucleotides. Okay, so let's get into this actual process. All right, so first we need to talk about the genetic code. How is it you get information from DNA and put it into, and make a protein from it? Okay, so remember proteins are made out of amino acids that are all linked together by peptide bonds. Hopefully that's old news by now. All right, and DNA is a triplet code, meaning you read it 3 bases at a time, and each one of those bases is called a codon. So for every 3 bases, you get 1 amino acid. So it's kind of like every 3 letters would give you 1 word in my previous awesome sentence about the fat cat. Okay, so 3 bases gives you 1 amino acid. All right, and then a specific sequence of DNA, not all the DNA, just a piece of it, that gives you the instructions for making a single protein is called a gene. Okay, so we talked about the protein insulin. The DNA in every single one of your cells has a little gene in there that's the instructions for making insulin, and the same goes for all your other 30,000 or so proteins that you can make. Okay, and each set of 3 bases is called a codon. That's more or less how that code works. You've seen this before. This is all of your amino acids. There's 20 amino acids, and you can get 64 different codons since there's 4 letters. 4 times 3, or I should say 4 times 4 times 4 gives you 64 possibilities. So those 64 codons will give you 1 of these 20 different amino acids. Unless it's a stop codon, then it's just going to make you stop. Okay, this is a chart you'll be using a lot this semester, and this is how we figure out which amino acid should go with a certain codon on the mRNA. Okay, a little overview of transcription. It occurs in the nucleus because that's where the DNA is. Remember, the DNA can't get out of the nucleus. It's stuck there. All right, we're converting DNA into messenger RNA, which is still information, but it's a different form of that information. Okay, it requires this enzyme called RNA polymerase. You've seen DNA polymerase before. That's what builds DNA. Well, guess what RNA polymerase does? It builds RNA. Okay, there's three main parts to this process called initiation, elongation, and termination. Okay, and this kind of shows you a little bit of the process here. And I'm going to discuss each one of these steps in a little more detail on the next slide. Oh, no, sorry. First, we have to talk about RNA polymerase as the enzyme. These are only found in the nucleus. You won't find them anywhere else in the cell because that's where transcription happens. It unzips the two strands of the DNA, kind of like helicase does in replication, but it does it all itself. It unzips the two, so it breaks those hydrogen bonds and pulls the two strands apart. Then it's going to move down just one strand, which we call a template strand, and it's going to add on the bases that match up with that. So this is kind of like half of replication happening, except instead of making new DNA, we are making RNA that matches the DNA, so it's complementary to it. Okay, you end up with what's called a transcription bubble in the DNA where it's been separated. Okay, it looks like this. This is our bubble right here, the transcription bubble. Okay, so transcription in a little more detail. First thing happens is initiation. The RNA polymerase binds to a specific sequence on the DNA called a promoter, all right? Like maybe you've heard of a concert promoter. That's somebody that works with bands, and they also work with the people that own the theaters and the stadiums, and they try to set up these concerts, all right? So they're called a promoter. Anyway, that's what the RNA polymerase enzyme binds to. After it binds, it starts moving down one strand of DNA and attaching nucleotides that match it. So it starts adding RNA that match the DNA. And as it does this, your RNA strand is going to get longer, which is where this name comes from. Elongation means to make something longer. Okay? Then after you're done with that, your mRNA, or I'm not sorry, your RNA polymerase is going to hit a spot on the DNA, and it's going to recognize that as its terminator, its stop sequence, and it's going to break away, and it's going to release the mRNA, which can now leave and go on to the next step of the process. Okay, so here are little diagrams that show you how it works again. I like this one especially. We have a double helix above and below. And we have a transcription bubble here. So the two strands have been unzipped, and this lighter blue strand here is the mRNA that's being added. You'll notice it's got U instead of T in it. So that is mRNA. And you'll also notice we're only copying one side. This side's not being copied. It's just there because DNA has to exist in a double helix in order to replicate itself properly. So that is transcription. Okay, and here's another look at this table, which we'll be looking a lot at later. All right, well, if you guys have any questions, let us know, and we'll see you next time. Thanks.